I have the great pleasure of chairing the Silver Medal Lecture. This award has been assigned annually since 1991 and recognises scientific excellence in the field of nutrition science. And over the past 29 years, the Silver Medal has been awarded to many outstanding nutrition scientists, and this year is no exception. I'm delighted that 2020 Silver Medal has been awarded to Dr. Vimal Karani. Vimal is an Associate Professor in Nutrigenetics and Nutrigenomics at the Hugh Sinclair Unit of Human Nutrition at the University of Reading, with whom I have had the great pleasure of working with over the past 5 years. He is an excellent scientist and teacher, a rare combination. And for the second year, Vimal has been nominated for the Reading University Student Union Teaching Excellence Award against very strong competition. Vimal is internationally recognised for his research in nutrient-gene interactions and cardiovascular disease-related outcomes, and he uses a combined approach from genetic epidemiology, statistical genetics, and molecular biology. He has received significant media attention for his work and is frequently invited to present around the world. One of Vimal's recent achievements is the initiation of a large-scale collaborative project called the Gene-Nutrient Interaction Collaboration, that aims to develop a personalised nutrition program based on the evidence from Nutrigenetics, Nutrigenomics, gut microbiome profiling, and dietary intervention studies using cohorts from various ethnic groups. It is this study that Vimal will be presenting on today. So it really gives me great pleasure to invite Dr. Vimal Karani, the successful recipient of the Nutrition Society's 2020 Silver Medal, to present. Thank you very much, Vimal.
Thank you, Julie. Let me share my slides. Are you able to see my slides?
Yes, we can see your slides, Vimal.
Okay, thank you. A very good afternoon to everyone. Thank you all so much for attending this session, and thanks to the Nutrition Society for giving me this wonderful opportunity to present the findings from my genuine collaboration, which aims to implement multi-ethnic population-based nutrigenetic studies of obesity in lower middle-income countries. Well, to begin with, what is a gene-environment interaction? In today's world, obesity has given to science a new dimension to delve into changing lifestyle factors, and a fast-forward urban outlook on life has resulted in a dramatic increase in the incidence of obesity worldwide. The advanced scientific technology that we have today has indicated that it all started at the beginning of life when the gene first comes into being. It's basically the interaction between the genetic and the lifestyle factors which contributes to the development of obesity. Genes load the gun, the environment pulls the trigger. This metaphor clearly explains the intricate interplay between the genes and the environmental factors in the development of obesity. So with this background, let me give you an overview about some of the basic aspects of genetics for those who do not have a background in genetics. So every eukaryotic cell has got a nucleus, and within the nucleus you have the genetic material, and the genetic material is nothing but DNA, and DNA is made up of nucleotides, and the 4 nucleotides are A, G, C, and T: adenine, guanine, cytosine, and thymine. And within the human DNA, we have nearly about 40,000 genes which have been discovered by the Human Genome Projects. And what do these genes do? The genes carry the genetic information for producing different proteins. And these proteins could be enzymes or secretory proteins or transport proteins or membrane proteins, basically performing various metabolic functions, which clearly shows that any defects in the gene or any genetic variations in the human DNA can have an impact on the quality and the quantity of the protein that is produced, and which in turn can have an impact on the metabolic functions. And that's why several studies have clearly demonstrated the role of genetic variations in conferring the susceptibility to obesity and various obesity-related outcomes. But what is this genetic variation? See, this individual has got this DNA sequence. You know that human DNA has got several genes, and every gene exists as 2 copies, and that's exactly what you're seeing as the 2 copies of the DNA sequence. Say, for example, this population has got 5 individuals, and you can see that 99.9% of the human DNA is identical, where 0.1% carries all the genetic variations. So what is that genetic variation? So if you look at this particular genetic variation, you can see that some individuals carry T nucleotide and some individuals carry the A nucleotide. So there's a change from one nucleotide to another nucleotide, which is what we call as a SNP, or a single nucleotide polymorphism. And depending on whether you carry T or A, you can classify individuals into 3 different categories. If you do not carry any copies of the genetic variation, those individuals are classified as 0 copy individuals. And individuals who carry 1 copy of the genetic variation and the other copy without the genetic variation, and those individuals are classified as 1 copy individuals. And those who carry 2 copies of the genetic variation, they are classified as 2 copy individuals. So for a given genetic variation or for a given single nucleotide polymorphism, you can have only 3 groups of individuals, 0, 1, and 2. And in general, 2 copy individuals generally have a higher genetic risk compared to 1 copy and 0 copy individuals. So I'm here talking about 1 genetic variation, 1 SNP. Our human DNA has got 3 to 4 million single nucleotide polymorphisms. And just imagine if you carry 2 copies in all these single nucleotide polymorphisms, which means that you have a very high genetic risk. So what is this genetic risk? So if you carry 0 copy, it means that you have low genetic risk, which means that you have decreased risk of developing a disease such as obesity. And you carry 1 copy, it means that you have a moderate genetic risk, which means that you have a moderate risk of developing the disease. And carrying 2 copies, which means that you have a high genetic risk, which means that you have increased risk of developing the disease. But just because you carry 2 copies, it doesn't mean that you're destined to become obese. You can still overcome this genetic risk by modifying your lifestyle. That is, consuming a healthy diet and doing more levels of physical activity can overcome the genetic risk of obesity. So this is the underlying basis on which we do all the nutrigenetics and nutrigenomics research. So with this background, let's move on to what is nutrigenetics and what is nutrigenomics. So nutrigenetics refers to the study of the impact of the DNA sequence variations on chronic disease outcomes such as obesity in response to a particular diet. For example, if I have a high genetic risk, which means that I'm carrying 2 copies and I'm consuming a high-fat diet, what is the risk of developing obesity? So in order to answer this question, I need to understand the interaction between the genetic risk and the dietary fat intake on obesity. So basically, I need to test for the interactions between the genes and the diet on obesity. And this study is what you call as a nutrigenetics study. Whereas nutrigenomics, as the name implicates, omics is expression, genomics is gene expression, and nutrigenomics is the impact of the nutrient on gene expression, how the DNA is converted to RNA and RNA to proteins, how a diet influences the gene expression. And findings from nutrigenetics and nutrigenomics are essential to implement personalized nutrition, where an optimum diet could be tailored according to that individual's genetic makeup. So let's have a look at what is nutrigenomics. Nutrigenomics refers to the study of the role of nutrients in gene expression. So it all starts with your dietary intake. The food that you're consuming has got all kinds of nutrients, macronutrients, micronutrients. So eventually all these nutrients need to be taken up by your cells. So once the nutrient enters into the cytoplasm, in the cytoplasm, it goes and binds to the specific transcription factors. And the main function of the transcription factor is to bind to the nutrient and take the nutrient to the nucleus where you have the DNA. So the nutrient binds to the DNA and nourishes the DNA and regulates the gene expression. That is, converts the DNA to RNA and RNA to proteins and proteins to metabolites. So the study of the expression of the RNA in response to a nutrient is what you call as a transcriptomics, and study of the expression of the proteins in response to a particular nutrient is proteomics, and the study of the expression of the metabolites in response to a nutrient is what you call as a metabolomics. And these 3 areas of omics approaches, they come under one big umbrella, which is a nutrigenomics. So understanding the differences in the gene expression level, which is what we call as a molecular biomarkers, understanding these biomarkers is very much essential for preventing the development of the disease by implementing personalized nutrition strategies. And this is how we translate the finding from nutrigenomics to personalized nutrition. While nutrigenomics is dealing with gene expression, nutrigenetics deals with the gene-diet interactions, which is going to be the focus of my talk today. So with regards to running all these gene-diet interaction studies or nutrigenetic studies, I'm involved in two large collaborative networks. One is basically the D-Cardia collaboration. D-Cardia stands for Vitamin D, Cardiovascular and Diabetes-related traits. This is being funded by the British Heart Foundation. To date, we have nearly about 35 studies comprising more than 100,000 individuals from different parts of the UK, US, and European countries. And with access to the UK Biobank, which has got 500,000 people from the UK population, and with consortia-based studies, and after excluding all the overlapping studies, we have a sample size of up to 1.9 million individuals. And these are some of the publications from the D-Cardia collaboration. So the D-Cardia collaboration is focusing mainly on developed countries. For developing countries, I've initiated this Genuine collaboration, which was initially funded by the British Nutrition Foundation, and Genuine stands for the gene-nutrient interactions. And these are the countries in which I've implemented the nutrigenetic studies. And what you see in the brackets are basically the funding organizations that have supported my research activities. So let's have a look at this Genuine collaboration. So it's been 7 years since I started this Genuine collaborative network. And the main aim of this Genuine collaboration is to implement nutrigenetic studies in contrasting ethnic groups, and which is actually one of the main missing gaps in the world of nutrition. We do have a lot of well-established cohorts with data on dietary intake, genetic data, and phenotypic measurements in developed countries, whereas such well-established cohorts were missing in lower middle-income countries. And with that objective in mind, I've started implementing the nutrigenetic studies right from the scratch of implementing a population-based study in each and every population. So it started off with implementing the first nutrigenetic study in the Indian population, then in Sri Lankan population, then Indonesian population, in Brazil, and in West African population, and West Asian, that is the Turkish population. And now we are currently running some nutrigenetic and nutrigenomic studies in the Peruvian population. And we have also submitted a few grant applications to explore the nutrigenetic studies in North African, South African, in Middle Eastern, Pakistani, Indonesian, and various other Southeast Asian populations. And in addition to all these ongoing collaborations in lower middle income countries, I'm also leading some of the gene diet interaction analysis in developed countries as well, including the UK, US, European countries, and the Australian populations. And these are some of the publications from the Genuine collaboration. So with regards to the success behind the Genuine collaboration, I would like to share some of the funding opportunities that I had as part of building up the Genuine collaboration. So it started off with the British Nutrition Foundation Drummond Pump Priming Award, which enabled me to establish this Genuine collaborative network. Then I got a series of all the British Council Newton funds to establish collaborations with various lower middle income countries, and from different parts of the Asian local research councils, I was able to establish the first nutrigenetic studies in various parts of Asia. And then I got the Newton Fund Researcher Link Award to implement the first nutrigenetic study in a Southeast Asian population, which is the Indonesian population. Then I got the Rank Prize Funds Nutrition Committee Award, which enabled me to travel to Australia, where I spent a period of about 3 to 4 months. And that helped me to get involved in a large grant, which is the MRC grant, which is an Australian grant. And then I got the GCRF funding to implement the first nutrigenetic study in the Ghanaian population. Then I got the local Brazilian Research Council funding to implement the nutrigenetic study in the Brazilian population. And last year, I got about £1 million award from the Medical Research Council to implement nutrigenetic and nutrigenomic studies in the Peruvian population. And now we have submitted a series of grants to look at the nutrigenetic aspects in relation to various chronic diseases, and also in relation to COVID-19 to look at the impact of COVID-19 in relation to the dietary intake. So given this background, let me go into some of the examples of the nutrigenetic studies that I've done in the developed and developing countries. So looking at the developed countries, so to begin with, it all started off with the discovery of the obesity gene. So this obesity gene was discovered exactly 13 years ago by two independent genome-wide association scans. One study was done in the UK population and the other study was done in the Spanish population. And both the studies clearly identified this obesity gene. So once this gene was discovered, people strongly believe that if they carry two copies of this obesity gene variation, they are destined to become obese. So to clear this fear from the minds of public, genetic epidemiologists decided to test whether consuming a healthy diet can overcome the genetic risk of obesity. And that was the time this genetic epidemiologist decided to test for the gene-diet interactions, and a lot of nutrigenetic studies started to come into picture. So, but what is so special about this obesity gene? Why FTO gene? The FTO gene is nothing but the obesity gene. FTO stands for fat mass and obesity associated gene, and the genetic variations within this gene have been shown to be strongly and consistently associated with obesity traits in various populations, in different ethnic groups, in men, in women, in adults, and also in children. In both these genome-wide association studies, we were able to find FTO as a top hit in all the scans. So which clearly identifies the fact that FTO is the strongest gene for obesity to date. But in all my nutrigenetic studies, in addition to the FTO gene, I'd also chosen several other genes which turned out to be the top hit in most of the genome-wide association scans, which included the MC4R and TMEM18, which are some of the most important genes, and today they are very good candidates for obesity. So we constructed a genetic risk score which takes into account of the combined effects of all these genetic variations. So we constructed a genetic risk score and we tested for the association with obesity and also tested for the interaction with the dietary intake. So let me give you some examples of the studies that I've done in developed countries. So just a couple of examples. So one is basically a study that we did in 11,000 European individuals from 5 different European countries, and we carried out a meta-analysis, and these individuals were followed up for 3.5 years, and which was done as part of the Diogenes consortium. So Diogenes is basically Diet, Obesity and Genes, which is a pan-European program that focuses on the impact of the genetic and the dietary factors in relation to weight gain. So here we found a very interesting interaction between the genetic risk and protein intake on weight gain. So in this graph on the x-axis, you find the protein intake, and on the y-axis, you find the weight change per year. The three lines that you see, they're basically high genetic risk, moderate, and the low genetic risk. And if you look at this figure, you can see that individuals consuming higher amounts of protein-rich foods, they alone showed a significant risk in gaining more weight if they carry a high genetic risk. So people who had high genetic risk, when they were consuming the excessive protein, and that was a time they were gaining more weight over a period of time. So which means that lowering the protein intake, that is less than 17% of the total energy intake, might be an effective strategy to prevent weight gain in those who have a high genetic risk, that is a 2-copy individuals. And another study which was done in the same cohort where we looked at the Mediterranean diet, and we know that the Mediterranean diet is considered to be the healthy diet because it's rich in omega-3 fatty acids, fruits and vegetables, and we would expect that consuming more amounts of Mediterranean foods will be able to overcome the genetic risk of obesity, and that's exactly what we had tested in this study. So in this graph on the x-axis, you find the Mediterranean diet score, which means that higher the score, higher the consumption of the Mediterranean foods. On the y-axis, you find the weight change per year. And the three lines that you see, basically individuals with low, moderate, and a very high genetic risk. And you can see that individuals having a high genetic risk, some of those individuals, by consuming higher amounts of Mediterranean foods, they were able to overcome the genetic risk of gaining more weight, suggesting that healthy diet can overcome the genetic risk of obesity. So the take-home message from the studies, nutrigenetic studies in developed countries, is basically the genetic factors and the dietary factors, they do not work independent of each other, rather they work together, interact together, contributing to the development of obesity. So which means that even if you carry 2 copies, it doesn't mean that you're destined to become obese. You can still overcome the genetic risk by consuming a healthy diet, which means that the healthy lifestyle can definitely overcome the effect of the gene on obesity. Now let's move on to the nutrigenetic studies in developing countries. So if you look at the developing country, it's even more a challenging situation because each and every ethnic group has got a unique genetic makeup and they have a unique dietary pattern. The geographical conditions are different. The levels of the physical activity varies from one population to another population. And there are no nutrigenetic studies to date. And without nutrigenetic studies, there's no future for personalized nutrition. So this was the main reason why I wanted to implement nutrigenetic studies in each and every ethnic group. So I'm going to give you some examples of the first nutrigenetic studies that were implemented in these lower middle income countries. So looking at the first nutrigenetic study that we did in the Asian Indian population, so this study was done in the population of Chennai, which is located in southern region of India. Here we looked at 2,000 individuals from the urban population, and we found a very interesting interaction between the genetic risk and carbohydrate intake on obesity. So in this graph on the x-axis, you find the carbohydrate intake in tertiles. On the y-axis, you find the odds ratio for obesity, that is the risk of developing obesity. And the red bar, those individuals with high genetic risk. And you can see that those individuals consuming high amounts of carbohydrate-rich foods, that is exceeding 500 grams of carbohydrate per day, and carrying a high genetic risk, they had 2.46 times increased risk of obesity. So given that the main source of energy comes from carbohydrate for the Asian Indian population, it is interesting to identify that we also found an interaction with carbohydrate intake, which is clearly highlighting the fact that reducing the consumption of carbohydrate might be an effective way to prevent weight gain in those individuals who carry a high genetic risk. And the second nutrigenetic study, that is the first nutrigenetic study, was done in the Sri Lankan population, which is another South Asian population. And Sri Lankans, of course, they have a diet which is almost the same as the South Indian population. They do consume significant amount of carbohydrates. And interestingly, we found an interaction with the carbohydrate intake again. So in this graph on the x-axis, you find the tertiles of carbohydrate intake. On the y-axis, you find the wasted ratio, which is an indicator of central obesity. And the red bar, the high genetic risk, and the white bar with dots is a low obesity genetic risk. And you can see that individuals consuming high amounts of carbohydrate-rich foods, exceeding 500 grams per day, and carrying a high genetic risk, they had significantly higher risk of central obesity compared to individuals with low genetic risk, and which again goes back to the same fact that reducing the consumption of carbohydrate is essential to overcome the genetic risk of obesity for this Asian Sri Lankan population. And the third study was in the Southeast Asian population, that is the Indonesian population. So we implemented this first nutrigenetic study in the West Sumatra Island in the city of Padang. So there's a very unique community of people, which is called as the Minangkabau community. Minangkabau is the world's largest matrilineal community, and this study was funded by the British Council and Newton Fund. And we looked at the Southeast Asian women living in this city. And here we found an interaction with a protein intake. So you know that the Southeast Asians, they do consume significant amount of meat products. So there's increased consumption of the animal protein within this population. And we found an interaction with the protein intake indeed. So in this graph on the x-axis, you find the tertiles of protein intake. On the y-axis, waist circumference, which is another indicator of central obesity. And the red bar is a high genetic risk, and the white bar is a low genetic risk. And you can see that individuals consuming low amounts of protein, that is around 70 to 75 grams of protein per day, and those who carry a high genetic risk. So what I'm trying to say over here is, if you carry a high genetic risk, you can still overcome the genetic risk of central obesity by consuming a sufficient amounts of protein, that is a low, the protein intake which has been mentioned in the lowest tertile. So 17 grams per day is not a low protein diet. It is indeed the dietary recommendations of the protein intake for the Indonesian population. So sticking on to the dietary intake or the dietary recommendations for the Indonesian population, that might be the effective way to prevent weight gain in those individuals who carry a high genetic risk. And interestingly, we were able to replicate this finding in another Asian population, which is the Turkish population. Of course, the kind of diet is almost the same. They do consume significant amount of meat products, and there is increased consumption of the animal protein as part of their diet. And we also found an interaction with the protein intake. So in this graph on the x-axis, you find the tertiles of protein intake. On the y-axis, you find the central obesity, and the red bar is high genetic risk. And you can see that individuals consuming more than 140 grams of protein per day and carrying a high genetic risk, they had 3.3 times increased risk of central obesity, suggesting that the protein intake, the protein consumption needs to be reduced to overcome the genetic risk of central obesity. And the last example that I'm going to give you is the first nutrigenetic study in the West African population. So here we carried out the study in the Ghanaian population, and we found an interaction with the fat intake, the total fat, and also with the saturated fatty acid intake. So in this graph on the x-axis, you find the fat intake. On the y-axis, waist circumference, again an indicator of central obesity, and the black bar, high genetic, and the white bar is a low genetic risk. And you can see that individuals consuming the total fat when it's exceeding 47 grams of total fat per day or exceeding 23 grams of saturated fatty acid per day and carrying a high genetic risk, those individuals had significantly higher levels of waist circumference compared to individuals who have a low genetic risk. So in terms of looking at the findings from the genuine collaboration, this genuine collaborative network has clearly established that there is some level of genetic risk for central obesity in different parts of the lower middle income countries. So the question is whether this genetic risk can be modified by a healthy lifestyle. And there are various dietary patterns that are existing in different parts of these lower middle income countries. For example, if you take the South Asian population, India and Sri Lanka, it was basically the high carbohydrate diet which was determining the genetic risk of central obesity. Whereas in the Southeast Asian and the West Asian population, it was a diet that was high in protein intake, a similar finding which was also observed in the South American population, which is a Brazilian population. When you're exceeding the protein consumption more than 140 grams per day, that was the time the genetic risk of central obesity is increasing as well. And for the West African population, it was basically the total fat intake and the saturated fatty acid intake. When it is exceeding 23 grams per day, then that increases the genetic risk of central obesity. So in addition to all these ongoing collaborations, now we have started exploring the Nutrigenetic and Nutrigenetic studies in the Peruvian population as part of the Young Life study. I'm just going to give you an overview of what we had planned for this Peruvian population. So this project is called as the SONGS project, the study of obesity, nutrition, genes, and social factors, which is being funded by the Medical Research Council, and it's being done in collaboration with GRADA, which is actually a private organization in Lima in Peru, and also collaborating with some of the investigators from the University of Oxford. So SONGS is basically an extension of the Young Life study, which is an international study of childhood poverty, focusing on 12,000 children in 4 different countries, which includes Peru, India, Ethiopia, and Vietnam. And all these children have been followed up in the last 15 years as part of the 5 rounds of data collection. Now we are planning to do the 6th round of data collection, and this 6th round is what we call as the SONGS project. And as part of this particular data collection, we are going to focus on both the classic markers and the novel markers of obesity. And the classic markers include the dietary intake, physical activity, clinical, biochemical, and anthropometric measurements. And as part of the novel markers, we are going to look at the genetic, metabolomic, and nutrigenetic markers. And also we are planning to understand the underlying genetic and the phenotypic differences that are existing in the sensory perception, that is the taste perception, aroma perception, your food choice, your food preference, and in addition to the social factors, which includes the physical places of residence. For example, having a fast food outlet nearby or having a supermarket nearby, does that have any impact on the prevalence of obesity? So basically, we are trying to make use of the classic and the novel markers of obesity. It is something just little more beyond than just a nutrigenetics study, where we are trying to dissect out various factors, etiological factors behind the development of obesity. So in summary, looking at the genuine collaboration, this provides the first evidence of all this gene macronutrient interactions on central obesity in all these lower middle income countries. So for example, if you want to implement a personalized nutrition strategy for the UK population, which is ethnically diverse, it's very important that we need to take into account of the finding from each and every ethnic group. All these nutrigenetics findings that have been implemented in these lower middle income countries are very much crucial in terms of being translated into a personalized nutrition strategy, which could be applicable for each and every individual living in the UK population. So I would like to briefly touch upon this particular paper, which is about the genuine collaboration, where I've discussed about various strategies and what are the plans for future plans in terms of implementing the nutrigenetic approaches in each and every other ethnic groups in lower middle income countries. So if you're interested in knowing it, it's available online. And one more thing, besides a macronutrient intake, the genuine collaboration is also focusing on the micronutrient intake, especially the vitamin D and B12. So there's always a very big question, whether is it the deficiency of vitamin D and B12 which is causing obesity, or is it obesity that is leading to vitamin D deficiency and B12 deficiency? Several epidemiological studies and several randomized control trials have been carried out. Still, it is under a big controversy. So I've used a Nutrigenetic approach to understand the relationship between the deficiency of these micronutrients and the development of metabolic disease. So basically, I looked at the impact of the obesity on vitamin D status and the impact of the low 25-hydroxyvitamin D concentrations on the development of obesity, looking at the Asian Indian population, a study that was done in 2,000 individuals. And so in this study, we found a very interesting interaction between obesity genetic risk and carbohydrate intake on 25-hydroxyvitamin D concentrations, where we found that obesity was leading to vitamin D deficiency through its interaction with the dietary influence. So looking at this interaction, so here on the x-axis, you find the carbohydrate intake in tertiles. On the y-axis, you find the vitamin D concentrations, and the black bar is high genetic risk, and the white bar is low obesity genetic risk. So if you look at those individuals who consume low amounts of carbohydrate and those who carry a low obesity genetic risk. So just because you carry a low obesity genetic risk doesn't mean that you can have higher levels of vitamin D. You can have higher levels of vitamin D only if you consume low amounts of carbohydrate-rich foods, which means that only if you consume a healthy diet, that is a low-carb diet, only then you will be able to have higher levels of vitamin D, even if you have some levels of genetic risk, that is a low genetic risk. And the second interesting finding is about the relationship between the metabolic disease and the vitamin B12 status, a study that was done in Southeast Asian population. So here we found a very interesting interaction between the vitamin B12 genetic risk and dietary fiber intake on HbA1c, which is a glycated hemoglobin, which is an indicator of type 2 diabetes. So in this graph, on the x-axis, you find the fiber intake in tertiles. On the y-axis, you find the glycated hemoglobin. The red bar is high genetic risk for B12 deficiency, and the blue bar is a low B12 genetic risk. So if you look at those individuals consuming low amounts of fiber-rich foods, which is an unhealthy diet, and carrying a high genetic risk for B12 deficiency, those are the individuals who had significantly higher levels of HbA1c. That is, those individuals who had a high genetic risk for B12 deficiency, they had a significant risk of developing type 2 diabetes if they consume a diet that is low in fiber. So which means that in summary, the obesity was leading to vitamin D deficiency through its interaction with the high carbohydrate diet for the Asian Indian population, whereas for the Southeast Asian population, it was a B12 deficiency that was leading to obesity through the influence of the dietary fiber intake. So these findings are very specific for these ethnic groups, because one of the most important reasons why I'm implementing the nutrigenetic studies focusing on each and every ethnic group is because the findings from one ethnic group cannot be applied to another population, because you are implementing that study for that ethnic group under the impression that, or under the assumption that you're going to develop a personalized diet based on these findings. So you cannot develop a personalized nutrition strategy based on the finding from another ethnic group. You need to replicate that finding in each and every population. So that is the basis of implementing these nutrigenetic studies in different ethnic groups. So there are plenty of nutrigenetic studies that are coming out from the genuine collaboration. So how are we going to translate the finding from nutrigenetics to personalized nutrition? So it all starts with the dietary intake. The food that you're consuming, it goes inside a body and it goes to 4 important compartments. One is basically where it is interacting with the human DNA, and that is what we have seen as a gene-diet interaction or a nutrigenetic study. And the second one is more about the nutrigenomics, where the diet is interacting with the metabolites, and that area of research is called as a metabolite diet interaction or the nutri-metabolomics. And the third important area is your gut lumen, where you have several microorganisms, not just bacteria, but also viruses, phages, and yeast. So the gut microbiome plays a very crucial role. So besides nourishing your DNA, you need to nourish your gut microbiome as well. And that area of research is the metagenomics. And most importantly, your non-genetic mechanism, which is the epigenetics, which is the epigenome. So your epigenome also needs to be nourished. That is, your diet can influence and regulate the gene expression. The epigenetics is nothing but the heritable changes in the gene expression, which is more towards the changes in the phenotype rather than the changes in the genotype. And the two important epigenetic modifications, which is the DNA methylation and histone modification, they entirely depend on the kind of diet that you're consuming. So in terms of looking at this translation of nutrigenetics to personalized nutrition, it is more important that we need to look at it as a four-way interaction, which is a metagenome, hyperbolome, epigenome diet interaction. And understanding this interaction is very much crucial for the implementation of the personalized nutrition, which is going to be the future in terms of preventing the development of chronic diseases such as obesity, diabetes, or even cardiovascular disease. So on this note, I would like to thank all my genuine collaborators and thanking all the funding organizations that have supported my work. And thanks to all the DCardia collaborators. And finally, thank you all so much. And thanks to the Nutrition Society once again for giving me this wonderful opportunity. And thank you once again.
Thank you very much indeed, Vimal, for that fascinating presentation. It really highlights the importance of personalised nutrition as a strategy for reducing cardiometabolic risk. As this is a award lecture, we don't have questions, but it's that time that it will give me great pleasure that I can virtually present you with the Silver Medal Award. I understand you've been sent the award, Vimal, so if you could show the award to us.
Yep.
So many congratulations in this award.
Thank you. Thank you so much, Julie. Can I take just a few seconds to thank a few people as part of getting this award?
Of course.
Yeah, sure. Thank you. And first of all, I would like to thank the Nutrition Society for recognizing this genuine collaboration, which is an important thing for addressing the missing gap in the world of nutrition. And thank you so much for that recognition. And I would like to thank all the genuine collaborators who have supported me. And also, I definitely need your support in expanding this genuine collaboration network. And of course, I would like to thank all my colleagues at the University of Reading who have supported me with this work. And thanks to my line manager, which happens to be Julie again. And finally, last but not least, I would like to thank my parents who have supported me. And I would like to dedicate this award to them. And thank you once again for this award. Thank you so much.
Thank you very much. That was lovely, a lovely presentation speech. So really that really leads to the end of this. Again, very many congratulations, Vimal, and this will conclude the silver medal presentation. And I hope to see you at the next presentation in a few minutes, which starts at 2 o'clock. Thank you very much.
Thank you very much.
Bye-bye.